Essential guidelines for pharmaceutical microbiology labs, ensuring product safety. Pharmaceutical microbiology laboratories play a pivotal role in the sterility testing of drugs, detecting and enumerating microorganisms, and testing bacterial endotoxins. These guidelines are crafted for any microbiology lab involved in pharmaceutical testing, whether independent or part of a manufacturing facility. They are based on existing guidelines and international standards, such as the International Pharmacopoeia, ISO/IEC 17025, and other WHO publications, ensuring a global alignment of practices. The document sets out to reinforce the importance of maintaining consistency in a laboratory environment, equipment, personnel, and testing methods to achieve reliable results that comply with regulatory requirements. 1. Personnel, key to laboratory success. Personnel are central to the effective functioning of a microbiology laboratory. The document emphasizes the need for competent and well-trained individuals to supervise and carry out microbiological testing. Personnel should have formal qualifications in microbiology or related fields and must undergo rigorous training before performing tests independently. Laboratories are expected to maintain detailed job descriptions, qualification records, and training histories for all personnel. Continuous training, especially in aseptic techniques, media preparation, and identification methods, is essential to maintain competence, particularly for tests that may not be performed regularly. The guidelines also stress the need for ongoing assessment and retraining as necessary, ensuring a high level of competence in handling microorganisms and maintaining laboratory safety. 2. Laboratory environment, ensuring contamination-free zones. The design and maintenance of microbiology laboratories are crucial to preventing contamination and ensuring accurate results. Laboratories should be dedicated spaces, separated from production areas, and equipped with adequate storage for samples, reference organisms, and media. Environmental control, including separate air handling and proper temperature and humidity regulation, is a key concern in ensuring sample integrity and reducing contamination risks. Laboratories conducting sterility testing should have designated areas that meet specific air quality and cleanliness standards equivalent to those used in pharmaceutical manufacturing. Environmental monitoring programs, particularly in high-risk areas such as sterility testing rooms, are required to track potential contamination sources. Regular cleaning, disinfection protocols, and controlled access to critical areas help maintain the necessary hygienic conditions in the laboratory. Revalidation of test methods, accuracy, and reliability. Validation of test methods is another major focus of the guidelines. Even for pharmacopoeial methods, laboratories must verify that the specific method used is suitable for their products and testing environments. Non-pharmacopoeial methods must be fully validated, taking into account parameters like accuracy, precision, specificity, limit of detection, and robustness. This section reiterates the need for rigorous validation, especially when testing samples that may inhibit microbial growth or interfere with results. This ensures that the laboratory's testing methods are both reliable and consistent across various. 4. Essential equipment, maintenance, calibration, and performance. Equipment used in pharmaceutical microbiology laboratories is vital for producing accurate results, and the document provides extensive guidance on the calibration, maintenance, and qualification of laboratory equipment. Each piece of equipment should have a documented schedule for maintenance, calibration, and performance verification, ensuring it remains within acceptable limits. For example, autoclaves used for sterilization must undergo validation studies to ensure temperature consistency and efficiency in sterilizing equipment and media. Detailed recommendations for maintaining and qualifying incubators, water baths, ovens, and other essential equipment are provided, ensuring laboratories maintain a controlled environment conducive to accurate microbiological testing. 5. Reagents and culture media, quality control practices. The quality of reagents and media directly impacts the outcomes of microbiological tests. This section addresses the sourcing, preparation, and verification of reagents and culture media. Media can be prepared in-house or purchased, but growth promotion tests must be conducted for each batch to ensure it can support microbial growth as expected. The guidelines stress the importance of handling media carefully to avoid contamination, recommending that media be stored appropriately and validated for performance, including recovery of target organisms and the exclusion of non-target organisms. 6. Reference materials and cultures. Reference materials and reference cultures are essential in maintaining the consistency and accuracy of microbiological tests. Laboratories are required to use internationally recognized reference strains to validate methods, verify media performance, and ensure the accuracy of results. The guidelines provide detailed protocols for handling, storing, and utilizing reference cultures, including recommendations for maintaining traceability and ensuring that reference stocks remain uncontaminated and effective throughout their use. 7. Sampling and sample handling. Sampling is a critical step in microbiological testing, and this section outlines the procedures for collecting and handling samples to prevent contamination or degradation. Samples should be collected aseptically using sterile equipment and stored under appropriate conditions until testing. Laboratories must have clear protocols for the receipt, identification, and storage of samples, ensuring that any issues with sample integrity are identified and addressed before testing begins. Proper sample handling minimizes the risk of erroneous results and ensures that testing accurately reflects the true microbial content of the product. 8. Disposal of contaminated waste. The safe disposal of contaminated waste is a vital aspect of laboratory operations. Laboratories must have procedures in place to dispose of contaminated materials in a manner that minimizes the risk of environmental contamination or exposure to laboratory personnel. These procedures should comply with national or international health and safety regulations and ensure that all contaminated materials are effectively neutralized before disposal.